Now, the viruses with small DNA genomes, as we have already studied the viruses, there are viruses present with very big genomes, and then the viruses present with very small genomes, and most of them are similar, genetically similar towards their host. So, the viruses with the very small genomes. First of all is the lambda phage M13. The M13 phage is the simple genome, very simple genome. It is about 6.4 kb in length, and it's the single-stranded. Most of the viruses which have very big, they are double-stranded DNA viruses, but the viruses which are very small, most of them are the single-stranded RNA, but this is not as a principle. It is positive sense single-stranded RNA virus, circular, but it is the circular DNA encoding about 10 genes. Only the 10 genes are encoding in M13 phage virus, and it is DNA virus, not the RNA virus. Then the M13 phage structure, how it's carry on, it's only have the about 10 genes. Icosahedral symmetry, it has the icosahedral symmetry, and the capsids can be expanded by the protein subunits. Of course, the capsid is made up of the proteins, the protomers or the capsomers, and then it is expanded by the protein subunits. And the genomic size can also be increased along with the capsid increase. Then the lambda phage. Lambda phage is the rapid packaging restraints are very important for the lambda phage. And the only DNA of between 95 to 110% of the normal genome can be packed inside the capsid of the lambda phage. It is about 49 kilobase pairs in length, in size, and the linear genomes, and they are the linear genomes, not the other genomes are present inside the lambda phage. And then the lambda phage, the substrate packed into the phage heads during the assembly, consists of the long containers of the phage DNA. DNA apparently willed by the phage head. Apparently the DNA will be willed inside the phage head of the lambda phage. Complete genome cleaved by the phage encoded the endonucleases. And the cause site, actually cause site is very important. Cause site is the site at the 5 end of the lambda phage, and it is the DNA ligase will cut it. We will see it in the diagram next, that how the cause site will act its action for the lambda phage replication, and after the replication, what is its function. T4 phage is about 160 kilobase pairs. The diagram shows the structure of the phage, T4 phage. It is double-stranded DNA having the linear genome in its structure. Then the T4 phage, the terminal redundancy is also very important, and it is also the similar with the cause site of the lambda phage virus, and the long containers of the DNA genome as similar as the lambda virus. We have already studied that it has the long containers of the DNA genome, and the genome is already willed inside the capsid, inside the head of the phage virus. Longer than the complete genome. Sometimes the genomic sequences becomes the longer as compared to the complete genome. Then the parvoviruses, linear, non-segmented, single-stranded, negative sense virus genome of about the size of the 5 kb. Most packed as the negative sense viruses, and the portion of the positive sense are very small, and 2 genes are very important, CAP and RAP. Both of the genes has different functions, and the parvoviruses we already studied today, are causing a number of diseases in the animals, in the mammals, and even in the human beings, just like the diarrhea and other diseases are caused by them. We will study them in detail later. Then the parvoviruses, the splitting pattern of each the genes, and the palindromic sequences for the hairpin structures are very important as far as the parvoviruses are concerned. Polyomaviruses. Polyomaviruses causes the very important disease, as maybe most of the Pakistanis know these days, that poliovirus and how the polio act, what is the function of the poliovirus, we will see in detail also. Polyomaviruses are double-stranded circular DNA, and they are also one of the smallest viruses present on the earth now, of the size of only 5 kb. DNA in the supercoiled form and 4 cellular histones are important, which are present inside the body, and the maximum information is only the minimum space is available with the polyomaviruses.